Hi, this is Deb, and I want to walk you through Chapter 3, the organic molecules of life. We're talking about huge molecules. They're really enormous, and most of them, not all of them, but most of them. And if you look at our list of the 4 groups, it's mostly what you eat. Carbohydrates, lipids, which are fats, and proteins. Then nucleic acids are your RNA and DNA. We got everything covered. Okay, so how are they made? They're made of monomers. Mono means one, mer means part. So these are the little subunits of one part that make up polymers. Poly means many, mer means parts. So these are like the boxcars of a train, and then we hook them together with specific bonds, and then we have a long chain of these, and we call that a polymer. And how are they hooked together? Dehydration. We take out a water molecule. I'll show you that probably way too many times in a few minutes here. And how are they broken apart? Hydrolysis. Water, lysis means to break. So we insert that water molecule again, and it breaks it apart. This is a video that's in the PowerPoint. You can watch it if you want. It's a nice one, but I'm not going to watch it right now because I have several pictures. Here's dehydration. You've got an OH on this side and H on this side. They come out and make H2O. And as they come out, these two hook together like the two boxcars of a train. But we want energy. We need to digest that food. We need to release that energy so we can run. We insert a water molecule, and it goes like this. And now those are released, and we can break them down further because they're kind of safer when they're in this larger arrangement. So one more time, same thing. All right. Carbohydrates. I never met a carb I didn't love, and that is so true. It's a universal energy source. Most have a ratio of 1 carbon to 2 hydrogen to 1 oxygen. We're going to talk about monosaccharides. 1 saccharide means sugar. And if you remember good old saccharin, that was kind of that was a good name because it really does mean sugar. Disaccharides. Di means 2 sugars joined together. Poly, many sugars joined together. So the first group of carbs, mono, 1 sugar unit. So these are the simple sugars. It's 1 sugar unit. And if you're filling out that table I attached, ready energy. That's the purpose. The carbon backbone, they can have 3 to 7 carbons, but it's really 1 unit. So when you write on the table, it's 1 monosaccharide, 1 monomer. They often have a hydroxyl group to make it polar right here. And that makes it soluble in water, which becomes very important. This is a silly video I put in. You can watch it, but it actually has some good stuff in it. All right, glucose examples is our main monosaccharide. When you talk about having low blood sugar, you're talking that the glucose is low in your blood and you're not accessing enough and you're feeling tired. The isomers of glucose are galactose and fructose. That means they have exactly the same number of carbons, exactly the same number of hydrogens, and exactly the same number of oxygens. They're just arranged slightly differently. But when we talk about it, we really are often just talking about glucose and sort of referring to the other ones. Very important biologically. It's the major source of cellular fuel. It's the last thing that's broken down to release ATP, which is energy, and that's why we eat food. Not because it tastes good, but because it releases energy. And like I said, it's what's transported in the blood of animals. There are 2 important pentoses, 5-carbon monosaccharides, deoxyribose and ribose. Do those sound familiar? DNA is deoxyribonucleic acid. RNA is ribonucleic acid. So those are the sugars that are used in DNA and RNA. We'll be talking about those in a minute. So our second group of carbs are disaccharides. And here's a hint to what's coming up. Once again, the purpose, if you're filling out that table, is energy storage. And this is a form in which sugars are usually transported. It's a bonding of 2 monosaccharides, disaccharides. Examples are maltose, sucrose, and lactose. So when we break those down, we break the bond that's hooking those 2 guys together. So maltose here, what's it used for? If you're a beer drinker, you're familiar with that. That's the sugar in malt, making beer. Sucrose is our table sugar. When you put your table sugar in anything, you're breaking that bond right there to release those 2 monosaccharides. Our sucrose often comes from beets, as a matter of fact. Here's another picture of dehydration and synthesis. Okay, we left off on lactose. So where do you find it? Milk. That's the sugar that's found in milk. And how is it different from sucrose? If you look over here, it's this bond right here that's different. There's sucrose, table sugar, and there's lactose. It has a beta bond, and this one has an alpha bond. Does it matter? Well, it matters a whole bunch if you're lactose intolerant. Turns out, if you're lactose intolerant, you cannot break this bond. And so you can't break it down, and it causes all kinds of crazy digestive issues, which I won't go into. All right, the third group of carbs, polysaccharides. So you're still filling out your table. We've got two types, and there's two functions. Energy storage ones, that's a little bit more longer term energy storage and structure. So we have many monomers hooked together. They're not as soluble in water. Why? Because this is we're going to store our energy. We want it stored and not breaking down before we need it. And because we need it to be strong if it's structural. So, filling out your table, plants store energy as starch. You're very familiar with that. Potatoes, corn, rice, all have lots of starch in it. And that's not for us, but it's for those organisms to have ready energy in the spring. We just tap into it. So starch molecules look like this. They're really long chains of glucose monomers. And here's a little teeny bit of one. You'll see it goes on and on. Then we have something called cellulose. We're talking structure now. And those monomers are bound by a bond that animals can't break. So if we can't break it down, why do we still eat it? It's called cellulose. And so celery, if you want to think of celery, it's mostly made of cellulose. Those long, stringy things in there are made of cellulose. We eat it because it's fiber and it's great for our digestive systems. It's the most abundant carbohydrate on Earth, and it's the most abundant organic molecule on Earth. 100 billion tons are produced each year. Whoa! If we can't break it down, why aren't we buried in it? Well, it turns out bacteria in the guts of certain animals, including termites, break it down for us. So that's how a whole ruminant works. They have several stomachs, which you've heard about. Well, there's one pouch with the bacteria in it that breaks those bonds for them. So it kind of goes through there. So they depend entirely on those bacteria to break cellulose so they can get energy. Special little trick of rabbits. They poo and then eat it again and then poo it out. So through that process, bacteria is introduced. Very disgusting. Poor rabbits. Okay, so animals, we have energy storage as glycogen in our livers. The storage and release is controlled by insulin, which you're familiar with. I won't tell the whole ski story, but it didn't go well for me. And so animals, structural, is chitin. That's in the exoskeleton of crabs, shrimps. And so like if you love shrimp and you peel the shell off, that shell is chitin. And we have discovered it has natural antibacterial and antiviral properties. So we're starting to use it in medicine. Pretty darn cool. And that's just a quick review. Which one is used by animals for energy storage? Which is used by plants? Purpose, energy. That's what I'm telling you. Fill that table out. You're ready to roll on this. All right, lipids. Let's begin. Fats. These aren't really a polymer, by the way, because they're made of different things. I think there's a question like that in the test. So they're insoluble in water due to hydrocarbons. Oil and water don't mix. That's what we're talking about. So hydrogens bonded only to carbon will not form hydrogen bonds. So you see that they have these long chains of carbon and hydrogen hooked onto a glycerol. So they're diverse, like I said, so they're not really polymers. Trick question, huh? All right, so we have the glycerol, and 3 fatty acids are hooked up in, oh, look, dehydration. And we have 2 different types, saturated and unsaturated. This guy up here is saturated, as this. Unsaturated is here. And it turns out that if there is an unsaturated one, that means that there's not a hydrogen on every bond and a couple of carbons have a double bond on it. It kinks. That lets it be liquid at room temperature, which is, I think, pretty awesome. So we're talking plants, olive oil over here. Saturated, packed tightly, solid at room temperature. Animal fats. Saturated fats from animals, full, packed, makes them solid at room temperature, and unsaturated, not full of hydrogens. There's some double bonds in there. They're liquid at room temperature, generally from plants, but also fish. Nearly all animals use fat over glycogen for long-term storage. Why? Well, we got to move around. And guess what? Fat weighs less. The only way a hummingbird right there can get off the ground is if their fuel, which they use copious amounts, because have you ever been around one? Oh my goodness. It has to be light so that they can take their fuel with them. So fat is good. It's not all bad. Purposes of fat. Insulation. This guy's heavily insulated against the cold. Polar bear, bottoms of their feet, as well as these little guys, are heavily layered with fat so that they don't freeze. All right, purposes of lipids. Phospholipids. This guy we'll hear more about next module. But what we have here are phosphate, which is polar, hooked onto two legs that are lipids that are nonpolar. Where do we use them? Oh, in our cell membrane. This is the cell membrane. It's two layers of those phospholipids. This inside part here is not water soluble. It's nonpolar. The out part is. So it's going to attract what you need in your cell, and then this acts as a barrier and a gate, this nonpolar area, and keeps everything in or out until you decide with these molecules what comes in and what goes out. So that nonpolar there is pretty important. More here, it controls what enters and exits the cell, just like I was telling you. So the polar heads are on the outside of this row, and the polar heads are on, well, the inside of the cell here and the outside of the cell there, with those legs hanging down. All right, well, let's talk steroids. Kind of an amazing thing. There are 4 fused carbon rings, and cholesterol here, let's talk about that one, is the precursor for our body to make testosterone and this type of estrogen. Now, I love this picture for a kind of a cool reason. Testosterone and estrogen cause very different changes in the human body. I mean, it's significantly different. And look, all that's different is this guy and this guy. This guy's not here, and this is different. Bingo. I think that's crazy. Those are the functional groups that make things do what they do. So, cholesterol has a bad rap. It can cause some health issues. So if you have a diet high in saturated fats and cholesterol, it can contribute to circulatory disorders. And so that's what we have here building up in here, that plaque that you've heard about before. And there's what we call good cholesterol, high density, and low, bad. The low stuff is what really adds to this. Another thing lipids do are the waxes for us. So plants make them waterproof. If you have any of those, like a jade plant or whatever these are, they're hydrophobic. That means they have this waxy exterior. They probably are in an area that is arid or dry, and so they need to retain all their moisture. So it's protective covering. Humans, we have earwax in here that to repel insects, which always creeps me out, and traps dust and dirt. So that earwax is supposed to continually come out. And then, of course, honeybees use wax for their structure. How do we use wax? We put it in candles, anything polishing, cosmetics, totally full of it. All right, ready for proteins. Proteins, we call them first place. That's what they're based on because they're the most important thing we think. 50% of dry weight of most cells are made of protein. So once you get the water out of there, we are talking protein. There are 6 main functions. We'll talk about each one separately here. Support. So that's the collagen for the ligaments, tendons, skin, hair, nails. Metabolism. Enzymes, man, you can't do anything without those enzymes. These guys, there's an active site on the enzyme. The substrate comes in, hooks on, and then through this interaction, 2 products are released. And so those kinds of reactions are important for life. Third group, transport. Those are those guys that go through the cell membrane that I showed you. These determine what enter and exit the cell. Defense. Antibodies. Bingo. It's the protein produced by the immune system in response to the presence of an antigen. So we have to identify it, and then we hook on an antibody, and then it can't hurt us anymore. The next one is regulation. So hormones, not all of them, some hormones are lipids, But the regulation of your body, several of them are hormones, are intracellular messengers, insulin and growth hormone. Growth hormone is a pretty important thing, and it turns out these pygmies in the Congo Basin have a malfunctioning growth hormone. It's not the right shape, and we'll see those things in a second, how that works. It's not the right three-dimensional shape, and so they don't grow past a certain height. The next one is motion. So it turns out proteins are these little guys that are contracting and making things move along using ATP at the smallest level with actin and myosin. And let's talk now what the building blocks are. So we talked about the purposes and what they do. Now, the protein monomer, one part, is an amino acid. And that's created out of an amino group, our carboxylic acid group, I cannot speak today, and then our carbon hooking it together. And so, what process is used to build these? Bingo! We take out water, same, dehydration. All right, amino acids are the building blocks. So there are 20 different amino acids joined by what? Dehydration to form a peptide bond. So a peptide are 2 or more amino acids, and a polypeptide is a chain of more. And the sequence that we put together, that first row, is what determines the 3D shape of a protein. This is all very concrete. The 3D shape makes it do what it does. So it's similar to letters of the alphabet that go together to make words, then go together to make sentences. Primary structure. Then, of course, we go to secondary structure when we have a pleated sheet or a helix. So they start pleating up and forming hydrogen bonds across. Remember, those are that can hook and unhook pretty easily. Well, this is how easily they unhook. Hot hair irons, like if you're straightening your hair or curling your hair, what that heat does is it breaks the hydrogen bonds in this 3D formation and puts it in, hooks together in the new form that you wanted, either straight or curly. Then tertiary, number 3. Now we have a globular shape, I can't speak today, going on. So instead of just hydrogen bonds, we now have covalent bonding between R groups. Enzymes do this, and that is why your body temperature and pH has to stay in a certain range, or your enzymes will uncoil, otherwise known as denatured. What's a good example of that? Egg white. Egg white's pure protein. Almost. And so when you crack it out of the egg, it's all gross looking and kind of mucousy. Then when you add heat, it turns white. What you've done is broken down, denatured it, added heat, broke all those bonds, and it uncoiled. And now it's a completely different product. And it will not do what it used to be able to do. So sometimes it goes to quaternary, the 4th level. Only very few do, but that means there's more than one polypeptide chain hooked together. But hemoglobin, that carries the oxygen in your blood, has 4 polypeptide chains hooked together and structured. So then you get to that level. Oops, sorry, I went too fast there. So you get to that 4 hooked together like that. Any of you who might have had chemical straightening or perm, there's some nasty smell stuff going on, that's a disulfide bond. That's permanent. And so on that level, they're breaking those quaternary bonds, reforming them, and then the neutralizing agent, and reforming them, and put them around a roller or a straight, and then put it on again, and you have permanently changed that structure, that 3D structure of the protein of your hair. Sometimes it doesn't go well. Protein folding diseases. We have these chaperone proteins that we think help, and if they get all messed up, then things don't work well. One that we're seeing is mad cow disease. And we also know some misshapen proteins are with cystic fibrosis. So here are some overall molecular shapes for protein. You can have helix here, or coils, or I'm playing with my fingers on the screen, or a pair of coils, or it can be even built up further, kind of like a cable, or you can have these globular shapes. So you can see how that goes. All right, now let's talk about nucleic acids. We're talking about DNA and RNA. And then we'll talk a little bit about ATP and ADP. All right, DNA is a genetic material that codes for everything in our body. Contained in the nucleus. And it's what makes up genes, which are the traits that are passed down in our chromosomes. RNA carries the directions for protein building from that DNA. So let's say you have a gene for blue eyes. The RNA carries that coding out to the ribosome where that protein that makes your eyes blue is built. You can kind of think of it as a factory line where the proteins are assembled. So here's DNA, it uncoils, messenger RNA matches the coding, takes it out, brings it on the ribosome, and these transfer RNAs bring in the amino acids and put them together in the right order. Here's another picture of that. So you have it uncoiling, the RNA gets the pattern, takes it out to the ribosome. We'll be talking about that a lot pretty soon. And then this transfer RNA picks up the right amino acid for the codon, the 3 little letters in a row, and brings it there and then hooks it on. So we end up with a very specific order of amino acids. This part's called transcription, this part's called translation. What's it made up of? Well, a phosphate. Here are the phosphate groups on the sides of the ladder. The sugar are on the sides of the ladder, so it's phosphate, phosphate, sugar, phosphate, sugar, phosphate, sugar on both sides of these ladders. And then the rungs are the nitrogen-containing bases. So that would be our A, T, C, and G, and there's complementary pairing that goes on. So how are they different? DNA and RNA together have adenine, guanine, and cytosine, A, G, C, and the phosphate. That's on the sides. These are the guys that are hooking together across the rungs of the letter. All right, DNA has thymine. That's traded out in RNA for uracil. I don't know why, but it is. And then in DNA, we have deoxyribose as a sugar, and RNA, ribose as a sugar. DNA is double-stranded. RNA is single-stranded. So we have those bases. Cytosine and thymine are the pyrimidine bases, and cytosine and uracil and RNA. So we see the uracil replaces thymine. The purine bases are the same. And so A always hooks with T, C always hooks with G. In RNA, A hooks up with U and C with G. And there's some really lovely safeguards that I adore that because it's those crazy 4 things that make all of our variety of traits. It's kind of crazy, I actually think. So we have this guy that's small hooks up with this guy that's big. Less chance for mistakes. This guy that's big hooks up with this one that's small. This one has 2 hydrogen bonds. This pair has 3 hydrogen bonds. So we have some safeguards because if these things get mixed up, that's when pretty bad things go wrong. And that's what I showed you again. That's how they're built. And so that coding right there is what's taken out here and makes these amino acids go in the correct order. All right, what's the relationship? All right, the functional group determines its behavior. When I was talking about the functional group on those estrogen and testosterone, that's just one little part that makes it do what it does. But on these, we're talking about amino groups and carboxyl. So now let's look at the big picture. Let's see how all of this fits together. So the relationship between proteins and nucleic acids is determined by the order of these amino acids. We have our sulfide bond there, and so the order of these and how they cross bond determines their 3D shape. And then that 3D shape of the protein that you see here, for example, determines its function. So it fits into a spot that fits it exactly. Some examples are, this protein channel here is a certain type shape protein, and that lets specific molecules go through, in and out of the cell. Here's another one. If you have one that picks up a certain molecule and shoots it through, it's this three-dimensional shape determined by the protein that makes it work correctly. So, how do we figure that out? How does that happen? It's our DNA that codes it. So when you talk about DNA, you're really talking about the coding for the amino acids that then are three-dimensional shapes that determine their function. They may determine your eye color, they may determine if your hair is curly, they'll determine your hair color. All of that is determined by the organization of these amino acids and how they turn into that three-dimensional protein. So we have the scripting here is carried out to the ribosome. The codons line up for those, and they match up A to T or A to U and C to G, and then each of these guys pick up a specific amino acid and bring it in and drop it off, and that's how they're hooked up in the correct order. Something goes wrong? Most of the time, a lot of the time, it doesn't matter, but sometimes a very tiny change can result in a significant illness. Here's an example. So here we have the coding, CAA, CTA, matches up with these, and then that codes for valine and histine and leucine, and it goes on across here, right? Those are the amino acids building the protein. Normal blood cell looks like this, like a little pillow-shaped thing. Well, if one is wrong right here, you see this? Instead of T and A, it's A and U. And it makes valine instead of glutine, I think that is, GLU. It makes one different amino acid hook in there, and then we end up with blood cells that are this shape. So it's a very crucial spot that cross-bonding puts it in the right space to make cross-bonding to make this perfect shape here of the three-dimensional protein, because this is created by the three-dimensional protein. And then this is with that bad coding going on. So it bonds differently, and it's the wrong shape. Now, a quick thing on energy. ATP, that's the energy we use. That's the final type of energy that we use in our body. Everything is converted to ATP. And guess what? It's made out of an adenine. Isn't that crazy? And then a sugar, and then a phosphate. So you have three phosphates hooked on, tenuous, 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 lots of energy to hook that last one on, and then we save our energy right there, and when we release it, that phosphate pops off, energy is released, and we can use it. That process between ADP going back to ATP and then back and forth is how we use our energy thousands of times a day. And, oops, sorry, I'd encourage you to watch that. And food for thought. Which molecule is more stable and why? ATP or ADP? Well, I told you that ATP is that one that takes tons of energy to hook that thing on. It's kind of like a set mousetrap, ready to spring and pop off. So the stable one that's not going to be springing and popping off is ADP. What type of molecule forms a peptide bond? Proteins. How is a chromosome related to a gene or a DNA? So we just talked about that. So the chromosome is made up of genes, and that's the DNA, and that's the ATGC business going on. What's the difference between a saturated and unsaturated fat? Saturated, solid at room temperature. It contains all the hydrogens you could possibly pack into it. No double bonds. Unsaturated has some double bonding, so it has a kink in there, which makes it liquid at room temperature. Like lard animal fat, saturated, unsaturated vegetable oil, like olive oil. And what's the name of the molecule for plant storage of energy and for plant structural support?